Hello everyone, myself Archana Haridas, Department of Biology, St. Mary's College, Thrissur. My topic is superantigens, haptens and adjuvants. Superantigens, they are the class of antigen which cause the activation of T cells and massive cytokine release. They are the microbial products have the ability to produce massive activation of immune cells, leading to the release of inflammatory mediators. Superantigens simultaneously bind and activating major histocompatibility complex class II molecules on antigen presenting cells and T cell receptors on T lymphocytes bearing susceptible V beta region. It is two types, exogenous and endogenous. Exogenous are soluble proteins secreted by gram-positive bacteria. Example Staphylococcus aureus and Streptococcus pyogenes. Endogenous, it is a cell membrane protein are encoded by certain virus that infect mammalian cell. Example mouse mammary tumor virus MMTV, Epstein-Barr virus EBV. Haptens. Landsteiner employed various haptens. They are small organic molecules that are antigenic but not immunogenic. Chemical coupling of haptens to a large protein called carrier yield an immunogenic hapten. Carrier conjugate. A hapten cannot function as immunogenic epitope, but when a multiple molecule of a single hapten are coupled to a carrier protein, the hapten become accessible to immune system and often function as immunodominant determinant. He immunized rabbits with a hapten carrier conjugate and then tested the reactivity of the rabbit immune sera with the hapten and closely related hapten coupled to a different protein. Landsteiner found that hapten plays a major role in determining whether it can react with a given antibody. Many biologically important substances include drugs, peptide hormones, steroid hormone function as hapten. Conjugate of hapten with large protein carrier can be used to produce hapten-specific antibody. Pregnancy kit containing anti-hapten antibody to determine whether the woman's urine contain HCG, which is a sign of pregnancy. Adjuvant are the substance that when mixed with an antigen and injected with it enhances the immunogenicity of that antigen. It is used when antigen has low immunogenic or small amount of antigen is available. Precisely how adjuvant augment the immune response is not known, but they appear to exert one or more following effects. Prolong the persistence of antigen, enhance co-stimulatory signals, induce granuloma formation, stimulate lymphocyte proliferation. Aluminium potassium sulfate, alum, is one of the example of the adjuvant. Prolong the persistence of antigen. Injection of alum precipitates slower the release of antigen from the injection site. So, effective exposure of antigen increase few days without adjuvant to several weeks with adjuvant. Free and water in oil adjuvant is also another example of the adjuvant. Freon's incomplete adjuvant contain antigen in aqueous solution, mineral oil, emulsifying agent such as mannate monosilate. Then antigen released slower from site. Freon's complete adjuvant contain heat-killed mycobacteria in water in oil emulsion, activate macrophages and make more potent than incomplete. Alum and freon adjuvants stimulate a local chorionic inflammatory that attract both phagocytes and lymphocytes. Other adjuvants stimulate non-specific proliferation of lymphocyte and thus increase the likelihood of antigen-induced clonal selection of lymphocyte. Thank you.